Hello students. So in the today's class we are going to see about the pollination. But before the pollination, just we'll do the revision of the yesterday's class, that is megasporogenesis. Since megasporogenesis is very important for 5 marks, for some of you have some of the students may have understood or some of the students may be still understanding. Just we'll do the revision now. Okay. So the definition says megasporogenesis is the process of formation of megaspore from the megaspore mother cell is called as what? Megasporogenesis. And this megasporogenesis starts from the archaeosporium cells. The megasporogenesis starts from the archaeosporium cells which are present near to the micropylar region. Okay, which are present near to the micropylar region. So this micropylar region is that means the cells which are present near to the micropylar region are dividing periclinally. They are dividing periclinally and they are going to form two cells. How many cells they are going to form? Two cells. One is called as outer parietal cell and the inner sporogenous cell. One is called as outer parietal cell and the inner sporogenous cell. Now till here you have understood? Okay. So inner sporogenous cell, later it is going to convert into a megaspore mother cell. Okay. Inner sporogenous cell, later it is converting into a megaspore mother cell. How? See here, this inner sporogenous cell undergo meiotic division. The inner sporogenous cell undergo meiotic division and they are going to form 4 daughter cells. Okay, what they are going to form? 4 daughter cells, and those 4 daughter cells are called as megaspore tetrads. What they are called? They are called as what? Megaspore tetrads. Okay, that means there is a presence of 4 daughter cells. So among the 4 daughter cells, one is functional. One is functional and 3 daughter cells are degenerated. Okay, one is functional and 3 daughter cells are degenerated. Okay, see, 3 daughter cells are degenerated. So see here, like this the 4 cells are formed here. Okay, like this. Yesterday I have shown you by drawing the diagram. It is a rough diagram, so you have to practice the diagram also. Okay, so this is the 4 daughter cells. Among the 4 daughter cells, 3 daughter cells are degenerated. That means they vanish, they destroy themselves and they are going to form only one. Their remaining is only one cell and that is called as megaspore mother cell. What do we call for that one functional cell? It is called as megaspore mother cell. What do we call that one functional cell? It is called as megaspore mother cell. What is that? Megaspore mother cell. Now, after this, you have to see the monosporic development function. Sorry, definition. What you have to see? Monosporic development definition. Now, what is the meaning of the monosporic development? Monosporic development means the development of the female gametophyte or the process of development of the embryo sac by the single megaspore mother cell. By the single megaspore mother cell is called as monosporic development. What is monosporic development here? That is the process of development of the female gametophyte. What is monosporic development here? Process of development of female gametophyte, or embryo sac. Okay, or it is embryo sac is called as monosporic development. Okay, monosporic development. So after the one megaspore mother cell formation, after the one megaspore mother cell formation, that is continuously undergoing a series of mitotic cell division. That is, 3 series of mitotic cell division occurs by the single megaspore mother cell. Imagine, see here, this is the single megaspore mother cell. Okay. This single megaspore mother cell undergo first mitotic cell division. Students, you people, it is not necessary to explain what is the meaning of mitosis. You people have studied in detail about the mitotic and meiotic cell division in the PUC first year. So mitotic cell division occurs in the body cells, okay, or somatic cells we will say, and it is equational cell division. So the one megaspore mother cell undergoes first mitotic cell division. So according to your knowledge, how many daughter cells has to be produced? 2 daughter cells, is it not? From the single megaspore mother cell, if it is undergoing one mitotic division, means it has to produce 2 daughter cells. Yes or no, students? Okay. Then this second stage, once again, it is undergoing the process called as mitotic cell division. Okay, once again it is undergoing the process called as mitotic cell division. And each cell, if they undergo mitotic cell division, then how many daughter cells are produced here? 4 daughter cells. Okay, see, each cells, if they undergo the mitotic cell division, then how many daughter cells are produced here? 4 daughter cells are produced here. So 1, 2, 3, 4. Okay, 1 parental cell, 2 daughter cell. 1 parental cell, 2 daughter cell. Okay, so here 4 daughter cells are produced. Then these individual cells, individual 4 daughter cell, once again undergo third mitotic cell division. Individual 4 daughter cell undergo third mitotic cell division and produces how many students? See, this single cell is there. From this single cell, 2 daughter cells are produced. Okay, this single cell is there. From this single cell, 2 daughter cells are produced. Okay, this one is there. Third one. So here from this, 2 daughter cells are produced. Fourth one, from this, 2 daughter cells are produced here. So totally how many daughter cells here? 8 daughter cells. How many daughter cells? 8 daughter cells. So these 8 daughter cells, after the formation of the 8 daughter cells, the wall formation occurs here. To each daughter cell, the wall formation occurs. As the wall formation occurs, they themselves are arranging into a female gametophyte. Okay, as the wall formation occurs to each cells, they themselves are arranging into a female gametophyte. See, 1, 2, 3. 3 cells, they are forming antipodal cells. Okay, so 1 cell from here and 1 cell from here. That is 2 central cells are produced. Next 1, 2, 3. So here there is a formation of an egg apparatus. Okay, here there is a formation of an egg apparatus. So egg apparatus is consisting of 2 cells. One is called as egg and the other one is called as what? 2 synergids. Okay, egg and the 2 synergids. And next here is central cells. You people know this one, central cell. Central polar nuclei we will say. 2 cells are arranged into central polar nuclei and these are the 3 antipodals. What they are? They are the 3 antipodals. So these 3 antipodal cells, as I have already told you, that antipodal cells are the larger cells and they are acting as a vegetative cell which provides the nutrition to the development of the zygote. Then synergids are called as helper cells or supportive cells, and below the synergids you can see the proteinaceous fibrous thickenings called as filiform apparatus. Okay, below the synergids you are going to see the proteinaceous fibrous thickenings called as filiform apparatus. So this filiform apparatus is there in the shark produced by the pollen tube. So pollen tube, when they travel and they just hit to the mouth of the ovule or to the micropyle region of the ovule, it is going to bear that shock and that is done by the filiform apparatus. Okay, so this is the brief revision of the previous class, megasporogenesis. Hope so in this class you have understood what is the meaning of the megasporogenesis and how the development of the female gametophyte occurs. Okay. Okay. So in the today's class, I have already told you we are going to see about the pollination and its types. Okay. So students, we have to see about the pollination definition first. Okay. So what is pollination? The definition says that transfer of pollen grains, transfer of pollen grains from the anther of the flower, from the anther of the flower to the stigma of another flower or same flower. Another flower or same flower. Pollination definition says that transfer of the pollen grains. So where the pollen grains are present in the anther to the stigma of the flower, another flower or same flower. Stigma is present in the gynoecium. Okay. So from the anther of the flower to the stigma of the another flower or of the same flower. That is called as pollination. What do we call? It is the pollination. Here we are going to see the types of pollination. You have understood the definition, general definition you have understood here. That is transfer of the pollen grains from the anther of the flower to the stigma of the another flower or of the same flower. Now we are going to see the types of pollination. So note down this point students, it is very important. So here we are going to see the presence of two types of pollination basically. Two types of pollination. One is called as autogamy. The first one is called as autogamy. Auto means self. Gamy means gametes. Male gametes and the female gametes. Autogamy, or we will say self-pollination. It is also called as self-pollination. Now, the self-pollination definition says that transfer of the pollen grain from the anther of the flower to the stigma of the same flower. Stigma of same flower. That is called as self-pollination. So, autogamy is also called as self-pollination. So, this self-pollination says that the transfer of the pollen grain from the anther of the flower to the stigma of the same flower, of the same plant body. Okay, or we can say within a single flower. Okay, that is the self-pollination. Here there is no much genetic variation we are going to see because the flower is of the one plant only. Okay, because of the presence of the same flower in the plant body, within the flower only the pollination occurs. We are not going to see much genetic variabilities. Okay, here once again the self-pollination is having a three conditions. Once again the self-pollination is having a three condition. The very first condition is called as homogamy. What is the meaning of homogamy? That is, the maturation of the anther and stigma occurs at the same time. What is homogamy here? Maturation of the anthers and the stigma occurs at the same time. That is called as what? homogamy. Once again I'll repeat the definition. See here, homogamy. It is the condition of the self-pollination. So homogamy means maturation of the anther and stigma occurs at the same time. That is called as homogamy. What do we call for that? Homogamy. Homo means similar. Okay, homo means similar. What happens here? Maturation of the anthers and the stigma occurs at the same time. Okay, that is a homogamy. The next condition is chesmogamy. Next condition is chesmogamy. Chesmogamy means what? That is the anthers and the stigma. The anthers and the stigma. The anthers and the stigma are exposed to the atmosphere. The anthers and the stigma are exposed to the atmosphere. That means to the atmosphere they are exposed, we will say. That is called as what? Chesmogamy. So example here is, we can give oxalis and viola. Oxalis and the viola are the examples of the chesmogamy. Last condition is cleistogamy. Now what the cleistogamy condition here? That is the flowers which is grown below the soil, or the seeds which grown below the soil are having the closed flowers. Okay, the flowers are not opened. The flowers are not opened, or we can say closed. The flowers are not opened, or it is closed, In this, what happens? Within the closed flower only, we are going to see the maturation. Within the closed flowers, anthers and stigma are present nearby. Very close. Anthers and stigma are present very closely. Anthers and stigma are present very closely, and as the maturation occurs, immediately pollen grain will go to the stigma. See, within the closed flowers, okay, within the closed flowers, the pollination occurs here. How? Here there is a presence of the pollen grain and the stigma. Pollen grain, when they mature, they go near to the stigma immediately, and then the self-pollination occurs here. So that is called as what? Cleistogamous condition. What do we call for that condition? Cleistogamous condition. The flowers are not open or closed, or they are closed. The flower is closed, or they are not opened. Within the closed flower, anthers and stigma are present very close to each other, and whenever the maturation of the pollen grain occurs, they go near to the stigma. Then the self-pollination occurs. The best example we can give is your groundnut seeds. Okay, the groundnut seeds is the best example for you people to understand what is the condition of the cleistogamous condition. Understood here? So this is about the three conditions, that is homogamy, chesmogamy and the cleistogamy. Before this, what we have seen? We have seen the definition of the pollination. What is pollination? Transfer of the pollen grains from the anther of the flower to the stigma of the same flower or another flower. Then we have seen the two types of pollination over there, that is the self-pollination and the cross-pollination. So self-pollination is also called as autogamy. What the self-pollination is also called? Autogamy. And here In the autogamy, we are going to see the homogamy, chesmogamy and the cleistogamy. Okay. So also we are going to see the bud pollination. So autogamy, that means self-pollination, cross-pollination, bud pollination. Basically two types are there, but bud pollination also is involved nowadays in the syllabus. Okay. Okay. So this, have you understood this one? Homogamy, chesmogamy and cleistogamy. Note down the definition and the examples here. Okay. Now we'll go for the cross-pollination. Okay. For what we'll go here? Cross-pollination. Next is cross-pollination. So this cross-pollination is also called as allogamy. The cross-pollination is also called as allogamy. Here what happens, students? Transfer of the pollen grains from the anther of the flower, to the stigma of same flower of the same plant body. Sorry students, it should be another flower. Stigma of the another flower of the same plant body. Stigma of the another flower of the same plant body, or stigma of the another flower of another plant body. Okay. So this is the definition of the cross-pollination. Cross-pollination is also called as allogamy. So here definition says that pollen grains of the anther are transferring to the stigma of the another flower of the same plant body. See here students, if we see the diagram here, this is, imagine that this is the plant body and here somewhere on the branches are present, the flowers are present on the branches. So here one flower is there, okay, here one flower is there. So the same plant body, two different flowers. So this is the cross-pollination. From here the pollen grains are transferred to this flower. This is one type of cross-pollination. And one more type is that two different plant bodies are there. Okay, two different plant bodies are there and two different flowers are there. So this is the one flower. Okay, this is the one flower and this one is the one flower. Okay, so here transfer of the pollen grains to the another flower of the another plant body. Here what happens? Transfer of the pollen grains to the another flower of the same plant body. That is, to understand this is called as what? Cross-pollination. What do we call for this? Cross-pollination. Got my point? So here once again in the cross-pollination two conditions are there. One is called as geitonogamy. One is called as what? Geitonogamy, and the other one is called as what? Xenogamy. Geitonogamy and the xenogamy. What is this geitonogamy? I have told you just that the transfer of the pollen grains from the one flower to the another flower of the same plant body. Here also it is just like a self-pollination only, but almost self-pollination, but no genetic variation is there. What is geitonogamy here? Transfer of the pollen grains from the one flower of the another flower of the same plant body, that is called as geitonogamy. Whereas xenogamy is nothing but the two different plants are there. Okay, xenogamy means what? The two different plants are there, two different flowers are there. So transfer of the pollen grains from the anther of the flower to the stigma of the another flower of the another plant body. See the difference of the geitonogamy and the xenogamy. It is the another flower of the same plant body and it is the another flower of the different plant body. It is called as geitonogamy and the xenogamy. So these are the two types of the cross pollination. Okay, what are the two types here? Geitonogamy and the xenogamy are the two types of the cross pollination. So once again I will repeat, concentrate here. Cross pollination can be defined as the process where there is a transfer of the pollen grains from the anther of the flower to the stigma of the another flower of the same plant body, or it may be the another flower of the another plant body. That is called as cross pollination. And this cross pollination is also called as allogamy. What cross pollination is also called? Allogamy. And here we are going to see the presence of two types of cross pollination. One is called as geitonogamy and the other one is called as xenogamy. Geitonogamy is nothing but the transfer of the pollen grains from the anther of the flower to the stigma of the another flower of the same plant body. That is called as what? Geitonogamy. Whereas xenogamy means transfer of the pollen grains from the anther of the flower to the stigma of the another flower of the another plant body. This is the difference between the geitonogamy and the xenogamy. Understood? So here we have completed the cross pollination. Okay. Next is we are going to see the bud pollination. Also we are going to see the bud pollination. Now students, bud pollination, it is just like a cleistogamous condition. So this bud pollination occurs within the closed flower, but those closed flowers are present just above the above the soil, or we can say aerial part of the plant body. Bud pollination means within the bud. Within the bud, maturation of the pollen grains occurs. Within the bud, maturation of the pollen grains occurs, and then the pollination is going to occur. Maturation of the pollen grain, and within the closed bud only they are reaching to the surface of the stigma. Okay, that is called as what? Bud pollination. What do we call for that? Bud pollination. So this completes your pollination process. So students, just a brief chart of the pollination we are going to see here. Just note down this chart, the pollination chart you have to note down. That is the pollination. Write down pollination, then self-pollination, that is your autogamy or the self-pollination, autogamy or the self-pollination. Then next is allogamy, or the cross-pollination, allogamy or the cross-pollination. And next is the bud pollination. Next is the bud pollination. Once again, in the autogamy, we are going to see three conditions. That is homogamy, chesmogamy, cleistogamy. Homogamy, chesmogamy, cleistogamy. Then in the cross pollination, two conditions we are going to see. One is called as geitonogamy and the other one is called as xenogamy. So this is the overall picture of the pollination. So what says pollination says? Three types, that is autogamy, allogamy and the bud pollination. Autogamy is also called as self-pollination, allogamy is also called as cross-pollination and the bud pollination. So here once again three types are there: homogamy, chesmogamy, cleistogamy, and cross-pollination are of two types. One is called as geitonogamy and the xenogamy. Okay, so this is the information about the pollination. Now students, you people have seen about the pollination and also you people have seen about the cross pollination. Now to overcome the inbreeding depression, see here, continuously if self pollination is going to occur, then what happens, the inbreeding depression is going to occur. That to overcome the inbreeding depression, some of the devices are used as the cross pollinating agents. Okay, so devices for cross pollination, devices for cross pollination. What happens from this? By the cross pollination, the inbreeding depression can be overcome here. Okay, so first one is dicliny, second one dichogamy, and in this dichogamy two conditions are there, protandry and the protogyny, protandry and the protogyny. The third one is self-sterility. Okay, what is the third one here? Self-sterility. Fourth one, heterostyly. And the fifth one is hercogamy. These are the 5 outbreeding devices. Okay, these are the 5 outbreeding. They are called as outbreeding devices. What these are called? They are called as what? Outbreeding devices or devices for the cross pollination. Devices for the cross pollination. How many are there? Dicliny, dichogamy. In that 2 are the protandry or protogyny. Then self-sterility, heterostyly and the hercogamy. So these are the agents or the devices which will encourage the cross pollination. What they will do? They will encourage the cross pollination. They are not allowing the self pollination to occur. What they are doing here? They are not allowing the self pollination to occur, but they are encouraging. Instead, they are encouraging the cross pollination to occur. Why the cross pollination has to occur? I have told you already. That is, genetic variation will be there and it can overcome the inbreeding depression. Okay, genetic variation will be there. Mutations we can see, changes we can see, new appearance we can see. All these points are there in the cross pollination if cross pollination occurs there. And also the inbreeding depression means what? The quality of the cereals and pulses, quality of the seeds can be improved over this by this cross pollination. Whereas by the self pollination, quality of the seeds can be seen up to few generations. First generation quality seeds quality may be best and second generation seed quality may be best. But as the generations are going to follow, as the generations are going to be continued, the quality of this